Hi everyone. First of all, before we really get into the week on COVID-19, I just wanted to say that I know this horrible virus pandemic has affected all of us in many, many ways. Life right now is way different than we would have imagined just half a year ago. And my sincere condolences to those of you who have had someone directly affected by this, whether they got ill and hopefully got better, or they um, eventually perished due to COVID-19. I know I lost my grandmother. She was in one of the nursing homes that became affected. So while we all have our own grieving, and I can't pretend to understand what any one of you might be going through, I have lost myself, so I do know how it feels to be affected by this COVID-19. So my prayers for all of you and my condolences for whatever direct effect this virus has had on your lives. We're not going to be talking too much about the epidemiology of COVID-19 or even the symptoms and kind of some of the physiology that it causes. We're going to focus more in this article on the cellular mechanisms by which viruses, COVID-19 in particular, kind of get into our cells, hijack our cells, and then replicate. And that is ultimately what really causes disease in many cases, as the cell replicates and negatively affects the cells it's infected, and then your immune system takes over. But I just wanted to talk a little bit about virus life cycle in general so that you can have a bit of a background before you go into this unit. I know that a lot of us have different backgrounds, so I just wanted to make sure we're on the same level. And then I'm going to talk a little bit about an article that used iPS cells to kind of model some of this, and then that should lead you well into the article that we're reading. All right, so COVID-19 is a virus that has a lipid coat. So there's a phospholipid bilayer around it, and then outside of that phospholipid bilayer, there sticks out these membrane proteins. These are kind of the spike proteins that allow it to bind to and infect cells. And then all of this basically encompasses the viral genomic DNA. And so the first thing that any cell has to do in order to replicate is gain entry into your cells, because this is a very simple genome. It doesn't have any of the mechanisms to replicate itself. It needs your cellular mechanisms to do that. So the virus will bind using its spike proteins to receptors on the cell, and this causes entry into the cell, but the cell and the virus also need to be able to fuse together in order for the genetic material to be released. So the article you're going to read focuses a lot on the receptor protein that the virus binds to, and then how this fusion event occurs, because otherwise, as this goes to the lysosome, this would just be degraded by the cell's degradation machinery. But if the virus is able to cause fusion with the phospholipid bilayer of your cell, then the genetic material becomes released, and that genetic material now basically can use the cell's machinery to not only replicate the genetic material, but also to make the viral proteins that this genetic code, genetics encodes. One of the genes, unfortunately for COVID-19, is actually even a gene that kind of helps shut off the translation, the making of your own proteins, so that your cell machinery will really focus on making the viral proteins. Those viral proteins, as they're made, they get stitched into the endoplasmic reticulum. This is the same way that your own proteins would be. If they're membrane proteins, they would be stitched into the endoplasmic reticulum. Those many proteins get made, and in the Golgi, they start to be put together. So the spike protein, all those other membrane proteins, starts to get put together along with some of the genome, and that basically causes new viruses to be packaged. And just as your secreted proteins would be, those go in vesicles that will fuse with the membrane, and now what was one virus particle entering can now be many, many hundreds of thousands of virus particles being released. So again, you have to have entry, you have to have fusion, you have to have the virus using the cell machinery to make more proteins, more of itself. Then you have to have packaging of the new virus particles, release, and then unfortunately they can go on and infect other things. So one of the other things I wanted to talk about a little bit is we just finished last week an article on induced pluripotent stem cells. And one of the real important aspects of induced pluripotent stem cells is the fact that you can now start to model disease. You can create human tissues that you wouldn't otherwise be able to have access to. And this can be really important for many reasons. One, it can help model diseases such as Alzheimer's and schizophrenia and a lot of neuronal diseases, for example, because you're not going to just take brain tissue from these patients. And even though you can get post-mortem tissue, that only shows so much. But another thing you can do is you can use those human cells or human organoids to start to test drugs. And so one of the articles that I didn't have you read, but it's basically using induced pluripotent stem cells to create engineered human tissues. Now, these aren't fully functional lungs or livers or anything like that, but they are tissue organoids that really represent the human tissue that SARS can infect. And so, in fact, these have created the vascular organoids. So one of the things that the COVID-19 virus can cause is blood clots, and also can affect kidneys. I haven't seen one that really shows creating organoids of the lungs, which would be really helpful. But by creating these from human-induced pluripotent stem cells, the researchers can now start to see what the effects of drugs are on humans, because ultimately we want to block human spread of this virus. And so they've essentially taken the genome of the COVID-19 virus, and they've started to predict what things might be able to block replication, block entry into cells. And I'm not going to go too much into all these things, but one of the things they decided is that if you take the receptor, which is ACE2, as you'll find out in the article you're reading, if you take a soluble version of that, you can try to block the virus from entering, because basically you start to arbitrarily bind to all those spike proteins that normally would block to the cell ACE if you have a soluble one that's not on a cell. But ultimately, they want to see if this really does work. So they create these human organoids. They take induced pluripotent stem cells. They de-differentiate the somatic cells. They re-differentiate them into whatever they're looking for. This one has a lot of blood vessels. It's the vascular tissue. And basically, when they start to add the virus to these human tissue organoids in the culture dish, they can look how much the infection happens. And so basically, if you add in the soluble receptor, they can see that they have reduced infection by quite a bit. And ultimately, this is kind of what your antibodies are doing. Your antibodies bind to the spike protein, and they can block infection as well. And then they did the same thing with kidney organoids. And so they basically show that these little organoids have all of the different cell types associated with the kidney, including nephrils and things like that. And once again, they show that adding more and more of the soluble receptor for ACE2 does appear to block viral entry. So the point wasn't really necessarily to talk about this, but I did want to bridge between the induced pluripotent stem cells and COVID-19. And induced pluripotent stem cells are indeed being used to make the human tissue organoids to basically test if we can find ways to block viral spread and viral infection. All right. So with that, I hope you enjoy reading the article that really talks about how we gained an understanding of the mechanism by which COVID-19 gains entry into our cells and then fuses with our cell membrane.